the last thing we need to do now is give you a little bit of background for the final lab, which is a week from now. So, and it's kind of a nice nice kind of send off into Thanksgiving because it's the digestion lab. I'm assuming most people are going to be kind of digesting either turkeys or some other stuff. There's lots of different things you can do without killing animals too, but turkey is pretty delicious. So let's talk about that. In fact, I should try to find, hold on one sec. It's kind of funny. But anyway. So for this last lab, we need to introduce a few basic concepts about digestion, but it's mainly about enzymes, which we already know lots about. So for the digestion lab, Prelab, that's what we're talking about now. We're talking about what do you need to know to write this digestion lab Prelab, which is something you should have done like a week from today. In general, digestion consists of two different things. One is the breakdown of your food. So, food is basically usually some other creature, right? It's a turkey, or it's a corn plant, or it's a yam or something that actually was some other living thing that you are now taking and you're chopping up and chewing up and swallowing. And it's made up of the same biomolecules that you need, like proteins and carbohydrates and fats and lipids, but assembled into a turkey or a corn plant or whatever. So the first thing you need to do is basically disassemble that creature into amino acids and monosaccharides and fatty acids and things like that. Right. So that's the first part of digestion, is take this thing, this food, which used to be some other organism. Even if you are being a vegan, you're still killing some organism, some plant that was growing and doing its own thing. Even if you're just eating seeds are little embryos of plants. They're actually alive in there waiting to germinate and become a new plant. So you're taking some other life form and you're repatterning its molecules into your own body using your DNA template, or else breaking down those molecules for energy to run your cells. So life is it's a little harsh when you think about it. It's just like things taking in other things and repatterning them and using them for their own needs. This is what we're going to focus on in the lab. The other part that we're going to talk about more next week is absorption, into your body. Right? Once you break down the food into amino acids and monosaccharides and stuff, you actually have to bring it across into your bloodstream. We've already talked about that to some degree with transepithelial transport of sugar into the blood from the intestines. We'll talk about that in more detail later. But this lab is going to look at breakdown of building block molecules. We're going to do what's called in vitro versus in vivo. What does it mean to study something in vitro?
Like in the fetus?
No, vitro, vitreous means glass. So it means in glass.
Oh.
Versus in vivo, in the living thing. So this means in test tubes and stuff, like try to recreate the conditions in the body, but outside the body in a test tube. And what's nice about that is then you have a lot of control. If you're trying to study something in an actual living animal, it's ultimately the most responsible way to get information about what's happening in the actual animal, but it's really messy and hard to control. So for in vitro, we're going to try to recreate the conditions of breakdown in different areas, like in your mouth, in your stomach, in your intestines, using different enzymes, but then we can easily adjust, like pH, for instance, and see how that affects things. So we're going to be doing it in vitro in glass. We're going to be studying breakdown. And the breakdown has two parts. There's mechanical breakdown. It's like chewing, for instance, where you're just actually physically ripping things apart. But that's not going to get you individual amino acids or individual monosaccharides. What do we need to actually break apart proteins to get individual amino acids, or break apart starch and get individual glucoses?
Saliva.
Say it again.
Saliva.
But what is insulin?
Enzyme.
Enzyme. Right. So the other part's going to be the chemical breakdown with enzymes. So that's what we're going to be looking at. We're going to be looking at three different types of molecules. We're looking at carbohydrates, at proteins, and at lipids. And we're going to be using different enzymes to break them down. And we're going to alter the conditions to see how that affects the breakdown. So let's see how this is going to work. We're going to have carbohydrates. We're going to have proteins. We're going to have lipids. In each case, we're going to have some, representative. For carbohydrates, we're going to have starch. You all know starch. Starch is just glucoses all connected together. For protein, we're going to use egg white. Egg white is made out of albumin. It's just a protein. For lipids, we're going to use just cream. Cream, which is fat, kind of suspended in some water a bit. So, and then each of these cases, we're going to need some kind of test for breakdown. For starch, we're going to have two actual tests we can use. We have Lugol's iodine. This is purple if starch present. It's kind of like yellow if not. Then we're going to have the Benedict's test. This is going to be different colors: green, orange, red, if there's maltose, or just, simpler sugars, right? What is maltose? Anyone remember what maltose actually is? It was like in the first week of class.
Is it glucose and galactose? No.
Glucose and galactose is lactose.
Lactose,
That is
yeah.
milk sugar. Maltose is just two glucoses together.
Glucose.
It's just a disaccharide with two glucoses. That's all maltose is. So if we have starch, with just a whole bunch of glucoses together, right? Starch is just a whole big long chain of if we have some enzyme, in fact, the enzyme we're going to have is called salivary amylase. It's a enzyme in your saliva that breaks apart the bonds between the glucoses. If we have salivary amylase coming in and starting to break these bonds, starch is going to start to disappear and maltose is going to start to appear. Right? So we have actually two tests that we've got going here we can both see if starch is not breaking down, then we'll know that that's true because Lugol's iodine will be purple and Benedict's, this is going to be blue if no simpler sugars. Or they're called reducing sugars. So if Benedict stays blue and Lugol's turns purple, what does that tell us about the starch breakdown? Was there any starch breakdown?
No.
And how do we know that?
It would be a different color if it was.
Right, so what would it look like if starch was breaking down? What would Lugol's look like? What would Benedict's look like?
Lugol's would
I'm not, whoever's trying to talk is not coming through.
Lugol's would be purple.
So if starch is breaking down, then there's not going to be any starch left. So what would Lugol's look like?
It would be yellow.
Yellow.
Stay kind of yellow or yellowish brown. And then Benedict's is going to then show up the presence of maltose. It'll turn green or orange. So when we get to the digestive system in more detail later, your digestive system is also looking both at the substrates of digestion, because it needs to know what is there, what kind of enzymes do we need to break this food down. It's also going to be looking for the products of digestion to see, are we done breaking things down? Are we getting the products? Do we have the substrates disappearing? So for the starch, we're going to have the salivary amylase, which is an enzyme from your saliva, and you will be able to then check if starch is breaking down or not, because you have a test to see, is there still starch there or not? You have a test to look, are there the products of starch breakdown, these simplest sugars showing up or not? So does that make sense? Yeah. For the proteins, we're basically going to take the egg whites and we're going to cut them into very regular looking squares, 7 millimeters per side. We're going to have every egg white square looking exactly the same when we start. How are we going to know if the protein is breaking down?
Measuring the size of it.
Yeah. So if the protein starts basically getting like disassembled into individual amino acids, which are going into solution, what's going to happen to the size of the square? I have a little square of egg white here, and it's starting to dissolve. What's going to happen to the size of this thing?
It's gonna get smaller.
It's going to get smaller. So if I see after a while it looks like this, that is evidence that this thing was getting digested. The protein is getting dismantled and going into solution. So that is going to be our test for the breakdown of the proteins. It's just seeing if the little squares of egg white are getting smaller. For the cream, we are going to use a pH indicator and mixed in with the cream, which is going to be litmus. So we're going to use litmus cream. Litmus powder mixed up with the cream. Litmus powder, this is blue if alkaline, turns kind of red, pink if acid. So when we first, or I should alkaline or neutral, I should say. So when we first mix the litmus powder with the cream, it just looks like blue cream. What does a fat molecule break down into? What was a triglyceride made out of?
Glycerol and fatty acid tails.
Exactly, glycerol and fatty acids. So what's going to happen if we start breaking this little puppy apart and freeing up the fatty acids? So now they're just on their own. What's going to happen to the pH?
It's gonna go down.
It's going to go down. It's going to become more acidic. Exactly. So, and that's going to make this litmus, this pH indicator, turn red or pink. So fat breakdown is going to make the cream start turning pink, because as the acids are freed up in the lipid breakdown, it's going to make things more acidic, and this litmus cream, which is normally blue if it's just kind of neutral, is going to start turning pink. So that's going to be our test for the breakdown of fat. Does this blue cream mixed with this litmus powder, which stays blue if it's alkaline or neutral, but turns red or pink if it becomes acidic. If fat breakdown's happening, acids are freed up, and then everything becomes acidic and it turns pink. So does that make sense? Kind of giving you this layout. We're going to have carbs, proteins, and lipids. And we're going to have these different tests that we can use to look at whether breakdown is occurring or not. So now I'm just going to look at the, oh, Sienna?
Just a question. Did we want to know what enzymes were responsible for the breakdown of proteins and lipids?
Yeah, I'm going to talk about that right now.
Oh, sorry.
Okay. So now what we're going to do is look at each one in detail. So the enzyme is going to be salivary amylase. Amylase are those things that break down starches. We're literally going to get it from the subject spitting in a little test tube and collecting it. It's just in your saliva. And its job is to break apart starches into smaller chains. Oh, then the other thing I need, hold on. Hold on one sec. There we go. Okay. So this is kind of showing the actual lab. So for carbohydrates, this first part is just kind of a control. If I take pure starch, what's going to happen if I add Lugol's to it?
Okay.
Yeah, it's gonna turn purple. It better. So this is, we're making sure that our assays work. If I add Benedict's solution for the pure starch, what will I see?
Green orange.
No. The
Should be
green
blue.
orange, it's going to stay blue. Green orange means that there are the simpler sugars showing up. So if it's just pure starch, I won't see that. It'll just stay blue. Whereas if I'm looking at like maltose in water, that is going to turn purple, green or orange with Benedict's, but it's gonna should stay kind of yellowish brown with Lugol's because there's no starch there. So if we look here now, if I look at starch and water, what do I think is going to happen? And I'm going to put them in a 37-degree water bath because that's mimicking the body temperature. So letting it kind of be at body temperature. So this is just starch in water. Do I expect starch to just spontaneously break down?
No.
Not really. What about if I mix up starch and saliva and let it sit at body temperature? What would I expect?
We'll start breaking down.
Yes.
Like we just said, the whole point, saliva has that amylase, that enzyme that breaks down starch. So we're going to see something very different probably. This one, it says add 5 drops of concentrated hydrochloric acid, before you add in the other stuff. What do we expect to happen in tube 3 here? What happens if you take some enzyme and you put it in this really concentrated acidic environment?
That enzyme's not going to work
Right? And why is that?
because it's
Or
been
denatured.
denatured.
Yeah, remember, that's again kind of going back to our themes from the beginning of the semester. If enzymes are optimized to work in a particular pH that they are, that they normally find themselves in, and when you take that pH out of that, they get all twisted differently, then they no longer fit their substrate and they don't work. So if in your mouth is normally pretty neutral. So if you take this enzyme, salivary amylase, which is optimized to work in the neutral pH of your mouth, and all of a sudden put it in extremely low pH, it's not going to be very happy, right? So does that make sense when you're thinking about what your prediction is going to be for tube 3 here? Any questions about that? No? So protein is going to be different. So pepsin is the enzyme that is working for the breakdown of protein in your stomach. And it works in your stomach. Just a few moments ago, we brought up pH in the stomach as part of innate immunity. What do we know about the pH in the stomach?
It's very acidic, the stomach.
It's very acidic. So this little dude is actually optimized. It's one of the enzymes actually that is designed, whatever, is optimized to work in really low pH. So which of these test tubes do you think is probably going to have the best breakdown? We have pepsin just with water, pure just acid, pepsin with acid, or pepsin with NaOH, a base.
Pepsin with acid.
Pepsin with acid.
Right, because this is going to mimic the environment that it's optimized to work with in the stomach. Right? It might have some activity if it's, if you start taking it all the way on the other side of which it likes to be in an acid environment and you make it basic, that's really going to mess with it. Right? So when you're thinking about your hypotheses here, think about protein breakdown. Again, this is happening in the stomach. That's the important thing to remember here. And in the stomach, it's naturally going to be very acidic. So that's going to make the ideal conditions for this pepsin to function. So that makes sense. For the lipid one, let me get an so lipid breakdown. Lipid breakdown gets a little trickier. There are lipases. In fact, we're going to be using pancreatin, which is a, whoops. This is a lipase, from the pancreas. But what is the problem? Let's say I have my pancreatin. I'll make it look like little wedges that can break stuff down. Actually, let's make it red. So pancreatin, we'll make it look like little axes. So here are my little axes that bash apart lipids. But what happens if I just eat a big pat of butter and it goes down into my digestive tract? What do we know about fats when you put them in the aqueous environments?
They don't dissolve.
They don't dissolve. No, they all just, think about non-polar things. If I have oil and water, it just kind of goes and becomes a big glob. So like this might be my oil. Let's say this is my olive oil I just ate, or my butter that I just ate, or whatever. I can have my pancreatin trying to break it apart, but it can't really get at most of it because most of it is in a big glob that is big hydrophobic puddle inside there. So I can shake it up. If I shake it up, what happens if you take oil and water and you shake it up?
Separates into little bubbles.
It just separates again. So we need some way to break up this oil or this lipid into small little things that are more accessible, but not have them all regroup right away. And the way that happens in your digestive tract is with bile. Bile is kind of like a phospholipid. It has one side that's polar, one side that's non-polar. So the bile molecules, they're called bile salts. Again, they have a polar side and a non-polar side. Amphipathic. So the non-polar goes towards the fat, the polar goes towards the water. So the bile salts do what we call emulsify. Emulsif. Gosh, can you spell? They emulsify the fat, means they allow it to stay in little droplets in the water without all regrouping. Does that make sense? So what happens actually in your small intestine, you are adding in both the pancreatin from the pancreas and the bile salts, which are coming from the gallbladder and the liver. And together, they can maximize the fat breakdown because now we have a lot more surface area to actually attack because the little droplets are staying separate, as opposed to this first condition where it was really hard to effectively break down the fats, even though we had the enzymes. So for fat breakdown, you need two things. One is you actually need an enzyme that can break down fats, but two, you need to emulsify the fat to increase the surface area so the enzyme actually can get at the fat molecules.
So does the enzyme essentially not start working until the fat's been emulsified?
Right. I mean, it can work kind of at the boundaries, but it can't really get at the lion's share of the fat.
Okay, so it's not like really effective, Max.
Right.
Emulsified.
So in fact, people who have their gallbladders removed, they are going to have to be much more conscientious about eating fatty foods because their efficiency at breaking down fats in their diet is way compromised. Because normally the bile salts are going to really make fat breakdown efficient. There might be some fat breakdown, but it's not going to be nearly as good if you don't have the bile. So that just takes us to our final condition in the lab here. We're going to have litmus cream. Again, litmus cream is just the cream with the litmus powder stays blue if the fat doesn't break down. It turns pink if the fat does break down. So we have litmus cream just with pancreatin, litmus cream just with bile salts, litmus cream with pancreatin and bile salts, and just litmus cream in water. Which one do you think is going to have the best breakdown?
3.
Yeah, again, we just talked about it. If you have the enzyme and the bile salts, that is going to be the optimal condition to break down the fats. How about, can bile salts break down fat on their own?
No.
No, they don't actually break anything down. They just emulsify. They keep the droplets separate, but they don't actually break apart the fat molecules. What about pancreatin on its own? Do you think it can break down fat?
No.
Just a small amount, right? Because you just said it can kind of get at the perimeter, but can't get.
Yeah, so it can break down fat, but it's not going to be nearly as efficient. Right, and the kind of a confounding thing here is cream is already slightly emulsified, right? Cream wouldn't exist. When cream separates, it's not emulsified anymore. But if the cream is staying as cream, it's already emulsified to some degree. So the bile is going to further emulsify it. So this is your kind of the same way you had a bunch of different hypotheses for this kidney lab. For this lab, for each tube, you need to have a prediction. So for digestion of carbohydrates, for tubes 1 through 5, what do you expect for the Lugols, iodine, and the Benedicts, and why? For proteins, what do you expect to see in terms of the breakdown of the protein, and as measured by the size of the square of the egg white, and why for each tube? For the fats, what do you expect to see in terms of the color, which is indicating breakdown for each of these 4 tubes, and why? So this is also going to be one of these kind of multi-part hypotheses. But the thing you can be kind of be all excited about as you're writing it up is knowing this is the very last lab of this semester. So there are no more pre-labs, no more Physiology 224 pre-labs like in your life even, hopefully.